Good afternoon, all. Good afternoon, Dr. Akanksha Mishra. I, Prabha Rajput, welcome you to this ISFCP dialogue series. So, today among us, we have Dr. Akanksha Mishra. She has done her M.Pharm from BBAU Lucknow and PhD from IIT BHU. Today among us, she will discuss about the Parkinson's disease, current treatment approaches, and future prospects. So, I welcome you, ma'am. Please proceed.
Thank you, Prabha. It feels an honor for me to be here with you all today. So, uh, first of all, I would like to wish good afternoon to everyone. So, here goes with the topic. So, the title of the today's online lecture is Parkinson's disease: current treatment approaches and future prospects. So, to know about the treatment approaches of any disease, first we should know what is the disease. So, to understand about any disease, it is a best to see it by the symptoms. So, what is Parkinson's disease? Basically, Parkinson's is characterized by various symptoms, which includes rigidity, reduced arm swinging, postural instability, gait, tremors. So, in the here in the screen, we can see there are two sections. In the left side pics, we can see the defective postures and walking patterns of Parkinson's disease patients. Whereas in the right side pics, we can see that the tremors, which are usual symptoms of Parkinson's disease, tremor reach to such an extent that it is very difficult for the patients to perform day-to-day simple tasks. So this is the basic representation of what Parkinson's disease is. Now we'll go to the theoretical aspects. What is the reasons for these tremor? So Parkinson's, it is a second most common progressive neurodegenerative movement disorder. Movement, because in the pics we have seen that movement is basically getting affected. And neurodegenerative means neurons are getting degenerated. And progressive, it means as the age progresses, the symptoms of the disease, they also get worsens. So basically there are multifactorial pathophysiologies involved. There are various factors for the disease to be occurred. But the most common theory, which is widely accepted, is dopaminergic theory. As per this theory, there is a progressive loss of dopamine neurons in substantia nigra pars compacta. So here comes the point. What is substantia nigra pars compacta? It is actually a part of the brain, anatomical part of the brain, that provide dopaminergic input to the striatum. Dopaminergic input is provided to the striatum, and basically dopamine, which is released into the striatum, and here after getting released, dopamine shows its effect after getting attached to the receptor. Here in the picture we can see, first of all, there is a human head. We can see the location of substantia nigra pars compacta, and then a simple pictorial representation of neuron. Then in the second picture, we can see there is substantia nigra, dark colored because of the presence of iron. And then in here we can see that in the second picture, there is a dark color has been diminished. Why? Because substantia nigra has been diminished due to the loss of dopaminergic neurons in PD patients. And as I have talked about their attachment to the receptor, that we can see in the third picture. So we can see here, first of all, there is a normal neuron and dopamine is getting released into the striatum. There already dopamine receptors are present in other postsynaptic neuron. And after getting attached to the receptor, dopamine is showing its movement, which is to provide basic normal movement to human body. Here in the second picture, we can see Parkinson's patients, the dopamine release is decreased. Receptors are present, but dopamine are less. So that is why the normal movement get hampered and movement disorder happens in Parkinson's disease patients. So this is the basic definition of Parkinson's disease, why it happens. Now we will go to next slide. What is special about Parkinson's disease? I mean, why the whole world is going after the research on Parkinson's disease? Reason is, first of all, the disease, the patients of the disease are growing rapidly. If we talk about our country, then there are more than 70 to 80 lakh people, 70 to 80 people per 10 lakh were suffering from PD in as per the 2016 data. Here, if we talk about different directions of our country, then we can see that in Parsis, the disease is highest. Why? The prevalence is highest because the Parsis are basically migrated community, uh, and there is a genetic disorder in them. So, due to this gene mutation, the disease is highest in Parsis. So, what is special about it? Again, because there are no cure is available till now. Only symptomatic treatment is available, but the basic problem, that is degeneration of neurons, it cannot be get treated till now. And here, this is simple some data about the number of patients. So we can see by 2030 in US, there would be around 1.2 million population suffering from PD. And in Asian countries where India is involved, by 2030, there would be 6.17 million people will be suffering from Parkinson's disease. So we can see that the number of people are high, and treatment, again, there is no treatment available to cure for the permanent curing of that degeneration of dopamine. So as per the title of my talk, first I'll let you know about the clinical management of PD. It means current treatment approaches. So we know that symptomatic treatment is available, means symptoms are getting cured. So how? Because dopamine is involved for these symptoms to be appeared. So there is the that is the reason why most of the drug treatments, they either increase the amount of dopamine in the brain so that more dopamine get attached to the receptor and show the normal movement. And apart from that, if there is any neurotransmitter in the body, we already know if there is any neurotransmitter in the body, it always gets metabolized. It always gets broken down by other enzymes to their other factors. So same happens with dopamine also. So other criteria to treat the Parkinson's disease is to decrease the enzymes that cause the breakdown of dopamine. So here in the table we can see that Parkinson's disease has been classified into three different stages. First is early stage, second is mid stage, and last one is advanced stage. So early stage has been further divided into first and second. So the treatment is available as per the severity of the symptoms. Early stage, there are mild symptoms, there are light tremors, and then further walking patterns, facial expressions, they are changed. Then the treatments are levodopa. Levodopa is the most common treatment for the clinical management of PD. Levodopa, as the name suggests, it is actually a dopamine precursor which gets converted into dopamine inside the body. So it is given. Apart from that, dopamine agonist, which gets attached to the dopamine receptor and shows the effects. And apart from that, MAO-B inhibitor. MAO-B actually is an enzyme which causes the degradation of dopamine. So to avoid that, monoamine oxidase B inhibitors are given. So mid stage, same treatments are given. Then further advanced stage. Advanced stage has been further divided into two different stages. The symptoms are severe, they are worst in this stage. It is basically impossible for patients to perform their day-to-day simple tasks, and it is impossible for them to live alone. They basically need 24/7 help from others people. So the treatment include, so we can see that there are some combinations of drugs because it will be difficult for any single drug to cure the symptoms at this stage. So there are various drugs like levodopa combination with agonist inhibitors, then carbidopa antacopa. Apart from that, there are some extended release drugs. Means it will be given once, but they will be released over the extended period of time. Apart from that, there are surgeries that include deep brain stimulation, where actually electrodes are inserted in the brain, and those electrodes, they actually act like the function of dopamine, so that movement disorders are basically cured. And apart from that, there are some anticholinergics. So this is the current treatment of Parkinson's disease. Now, as per the topic, future prospects. Future prospects, if we talk about, because there are no cure is available till now, only symptomatic treatment. So first of all, we should know what is disease modification. Disease modification is actually related to the neurons which are getting degenerated. So it is classified into two categories: neuroprotection and then neurorestoration. Protection means the underlying degeneration of neurons to slow down this degeneration. Restoration, after getting this degeneration slowed down, now the task is to regenerate these neurons, which is actually impossible till now. So disease modification, if we talk about the future prospects of Parkinson's disease, so it is still a dream. So various research have been performed for this, but still clinical trials have been failed. But there are some drugs which are in the phase 3 clinical trial for attaining this disease modification of Parkinson's disease. Because we talked about clinical trials, so let me show you some examples how these clinical trials have been failed, how different mechanisms have been involved, but in that case also the clinical trials have been failed. The reason, basically the reason is either they have got inconclusive results, means they cannot get any results, any exact results. Reason, because symptoms were getting worse. So the basic problems with Parkinson's was still there. So if we see these drugs, first of all, there is pioglitazone. Again, phase 2 trial failed. Then other drugs, for example, coenzyme Q10, which is an antioxidant and bioenergetics. Again, phase 3 trial failed. Then creatine, again, mitochondrial bioenergetics, it is used. So phase 3 trial failed. Then there are various drugs named as SCH420814. This is adenosine receptor antagonist. Adenosine receptor actually decrease the release of dopamine. So antagonist was given, but again the drug was failed in phase 3 clinical trial. Then riluzole was given, glutamate antagonist. Glutamate action is similar to decrease the release of dopamine, but again after giving its antagonist also, the clinical trial was failed at phase 3. Apart from that, various compounds, various drugs like NADH, melatonin, ropinirole, pramipexole, which are dopamine agonist, and then salicyline, rasagiline, which are MAO inhibitor, they were given to the patients, but again they were failed in clinical trials. So we can see there is highest number of failures to achieve this disease modification. That is the reason disease modification is still a dream for Parkinson's disease patients. So it is the one reason why a whole world is coming together for to get the cure of this Parkinson's disease. So what is the need of the hour at present? Because we can see that number of patients are getting increased day by day, but neuro restoration means to regenerate those dopaminergic neurons, it is still a dream. So we have to come together as a community and try to find the cure of this Parkinson's disease. And people are doing that. There are various clinical trials which are under the pipeline, and we are getting the success also. But still, there is no marketed drug which can through which we can achieve this neuro restoration. So we will also talk about a little bit about the pathogenesis of Parkinson's disease. What is the pathogenesis? Means the etiology. Etiology means the reasons which are involved. So the complete etiology is still under research, because the Parkinson's disease is sporadic disease, means the exact causes is still unknown. But there are various factors which are involved in it. First of all, pathogenesis means energy metabolism and aging. As the age goes on, the energy metabolism decreased in subjects, any normal subjects. So that act as a factor for the PD to happen. Apart from that, impairment of ubiquitin proteasome system. This system is involved in the degeneration of that or we can say dysfunctional protein. But as the age goes by, or in PD patients, there is impairment of these systems. So that is why what happened that those subjects are prone to PD. Then genetics, there is several genes are involved in the Parkinson's disease. For example, alpha-synuclein. Due to genetic mutation also PD happens. Then there are some environmental triggers. Environmental triggers, for example, there is one pesticide rotenone. Rotenone is actually mitochondria complex 1 inhibitor. So mitochondria complex 1 inhibitor, it actually causes the degeneration of dopamine. So it is directly related to Parkinson's disease. Then mitochondria dysfunction, because it is the powerhouse of cell. So any dysfunction in mitochondria will lead the cell death, and ultimately there will be loss of dopaminergic neurons. And then oxidative stress. Oxidative stress is mostly related to most of the disease. It is the very simple phenomena that in our body there is a balance between antioxidant system and oxidative stress. But as the age goes by, or in PD patients, there is imbalance between these two, oxidative stress and antioxidant system. So when oxidative stress dominates this antioxidant system, there are chances for the PD to happen. Apart from that, there are some enzymes all involved. Glucocerebrosidase, that is lysosomal enzyme. So this is also an important factor for PD pathogenesis. And then alpha-synuclein pathology. Alpha-synuclein also cause the mitochondrial dysfunction. Apart from that, it is an important part of Lewy bodies which are found in the brains of PD patients. So all these factors are responsible for PD, and by targeting any of this factor or any of the linked pathways, neurorestoration may be achieved. So multiple pathways are being targeted these days, and effects are ramping up to find the cure. Uh, Now, as I have talked about some pathogenesis, so I'll just want to let you know in the pictorial representation how these factors are actually involved in causing the PD. So we should see it from the last so that you can understand it better. We know that Parkinson's disease is responsible for motor deficits. It means there is a loss of uh, dopamine, due to which motor deficits happens. So ultimate result is this. Now from where the picture starts, 6-OHDA. First of all, we should understand what is 6-hydroxydopamine. 6-hydroxydopamine is actually a toxin. It is actually a mitochondrial toxin. Here in the figure, we can see that it is actually injected in the body. So we'll talk about it later. First, we'll go here. So 6-hydroxydopamine, it is when it is injected in the rat to mimic that PD symptoms. So it is actually attached to dopamine transporter and goes to dopamine neurons. Here it undergoes auto-oxidation and directly affect mitochondria. So mitochondrial dysfunction happens. Apart from that, because auto-oxidation is happening, so hydrogen peroxide and reactive oxygen species, they are getting generated. And at the end, they are causing oxidative stress. Both 6-hydroxydopamine and oxidative stress collectively cause the mitochondrial dysfunction. So when mitochondrial dysfunction happens, there is a release of cytochrome C. Are actually two pathways for the mitochondrial mediated death. So there is one is internal and other one is external, intrinsic and extrinsic. So where the mitochondria is involved, that is intrinsic pathway of apoptosis, cytochrome C is released. That is intrinsic pathway of apoptosis. Apoptosis means cell death. So then if cell death happens, the dopaminergic cell death is happening, then striatal dopamine release will be less, dopamine transporter will be less, and tyrosine hydroxylase, which is the basic enzyme for the production of dopamine, it also gets decreased, and at the end Parkinson's disease happens, which is characterized by motor symptoms. So here I have attached one more factor, that is nuclear NRF2. What happens when oxidative stress happens in the body? Naturally, nuclear NRF2, it gets activated, and then nuclear NRF2, it causes the release of antioxidant system that includes various enzymes, for example SOD, catalase, and reduced glutathione. So these three enzymes basically makes the antioxidant defense system, and it decrease the oxidative stress, and when oxidative stress will be decreased, mitochondrial dysfunction will be itself decreased, and at the end, by following this cycle, Parkinson's disease may be cured. And apart from that, nuclear factor NRF2 also affect alpha-synuclein oligomers, because we know that Lewy bodies are the basic character of Parkinson's disease, which is found in the brains of PD patients. So nuclear NRF2, it also decrease the alpha-synuclein oligomer, through which PD can be treated. And alpha-synuclein oligomer, how it affects mitochondria function? because it decreases the mitochondrial protein import. So at the end, if mitochondria will not get its proteins, it will ultimately be dysfunctional. So this is the basic cycle through which I tried to let you know about these factors which I discussed in the previous slide. So I hope you got the gist of this slide. So 6-hydroxydopamine we'll just talk about a little. So this is actually a toxin through which we can mimic the symptoms of Parkinson's disease in rats to perform the research. So here we can see the figure of a stereotaxic injector. Here, after first rat was anesthetized using any anesthesia. So after that, the fur of the head was shaved, and then a cut was made in the scalp, and then we found the bregma point, and then there was one Hamilton syringe which was attached, we can see here, which was attached here, and then a hole was drilled, which was from bregma point. It was taken from different coordinates, and then 6-hydroxydopamine was injected using Hamilton syringe. Using this procedure, after 4 days the PD symptoms starts to appear, and after 28 days of this surgery, the PD symptoms are at its severe. So the whole pathophysiology, the whole symptomatic procedures of a PD, it can be reproduced. So this was the basic toxin which is generally used to mimic the PD symptoms. So here is all. Here we discussed about the current treatment and what can be done in future. So thank you. Here is the end of my slide.
Thank you, Dr. Akanksha Mishra. It was a very wonderful and informative lecture. I'm sure it will help to our students and other researchers who are working on this Parkinson's disease. It was very informative. Like they can, after listening this lecture, they will get to know the scope of like the Parkinson's disease, where they can work by modifying the several drugs which can be promising the anti-Parkinson's like symptoms or effects. So I'm sure it will be very helpful to all of you guys. So it's open to students. If you have any question, please ask. It is for like 1 or 2 minutes open discussion. Dear students, if you have any question, please ask.
I hope everyone got the introduction for Parkinson's disease.
Yes,
And if
ma'am.
if you have any question, you can contact me anytime. I'll share my email ID with Dr. Prabha.
Yes, if students have any questions, they also can drop their message in the chat box. So it's okay, ma'am. I think they don't have any question for now. Thank you so much, ma'am.
Okay, thank you, Dr. Prabha. Thank you.
Thank you.